The category of especially dangerous infections of a bacterial nature includes plague, tularemia, cholera. The first two are zoonotroponosis. Only people get sick with cholera. Especially dangerous infections, SDIs, are a group of acute contagious human diseases that, capable of sudden appearance, rapid spread, and wide coverage of the population, are highly contagious in small infectious doses, are transmitted by various transmission mechanisms, cause severe diseases, capable of causing epidemics and pandemics, cause a high percentage of deaths. Studies are carried out in the most isolated laboratory, biological safety level 4, high individual and public risk. World Health Organization pathogenicity classification. Plague is known as the Black Pestilence or Black Death, an acute natural focal infectious disease. Anthropozoonosis is characterized by severe intoxication, fever, skin lesions, lymph nodes, lungs, the ability to take a septic course, accompanied by high mortality and the possibility of epidemic spread. It is a zooanthropogenic natural focal bacterial infectious disease. Its etiology. The causative agent of the plague is the plague bacillus, Yersinia pestis, discovered in June 1894 in Hong Kong by the Frenchman Alexandre Yersin and the Japanese Kitasata Shibasaburo. Its source is a sick person or a sick animal. Taxonomy and Morphology. The causative agent of the plague belongs to the family of Enterobacteriaceae, the genus of Yersinia, species of Yersinia pestis, which is the causative agent of the plague. Morphology. Yersinia pestis is an ovoid, barrel-shaped, non-motile, gram-negative rod. Plaque 1000 form spores. A characteristic feature of the plaque microbe is a bipolar color. Bipolarity is especially pronounced in smears prepared from human or rodent organs. It is well detected when stained with methylene blue, according to Leffler. Has a delicate capsule that protects bacteria from phagocytosis. Yersinia pestis has several antigens. Antigens F1 and F2 is the main component of the surface structure of bacterial cells of a protein nature. The V antigen is also a protein. W antigen is a lipoprotein complex. These antigens are associated with the cell wall. Yersinia pestis has crossed antigens with other Yersinia and Enterobacteria, as well as with O group human erythrocytes. Pathogenicity factors. The virulence of plaque pathogen is associated primarily with its adhesion to the epithelial cells of various organs and tissues, depending on the entrance gate of infection. The capsule and surface structures of the cell wall are involved in adhesion. Various enzymes and toxins produced by these bacteria are involved in invasion and aggression, such as suppression of the phagocytic activity of macrophages. Significant importance in the pathogenicity of Yersinia pestis has a mouse toxin, which blocks the functions of cellular mitochondria of the liver and heart, and also causes the formation of blood clots. Mouse toxin refers to protein toxins tightly bound to the bacterial cell, the synthesis of which is controlled by a plasmid. A certain role in the virulence of plaque bacteria is played by such aggression enzymes as plasma coagulase and fibrinolysin, localized in the outer membrane of the bacterial cell, and endotoxins. The microbe is a facultative anaerobe. It grows on a simple nutrient media, such as meat peptone agar, meat peptone brose, Hottinger agar, Martin agar at pH 6.9–7.2. The optimum growth temperature is 27 to 28 Celsius degree, but can grow in the temperature range from 2 to 40 Celsius degree. The capsule is formed when grown at temperature of 37 Celsius degree. virulent strains form R forms. On liquid media, after 48 hours, a delicate film is formed, from which threads that are stalactites descend. At the bottom there is a loose sediment. The broth remains transparent. Shaking causes agglutinogenic growth. To accelerate growth, growth stimulants are added to nutrient media such as sodium sulfite and blood. When growing on solid nutrient media, after 8–12 hours, grayish mucous microcolonies appear with jagged edges, in the form of broken glass. After 18–24 hours, the growth of colonies with raised edges resembles a lace handkerchief with yellowish or brownish color of the protruding central part, the main virulent R type. In the English literature, the colonies are described as hammered copper, which when aged take on the appearance of fried eggs. After 48 hours of cultivation, large colonies are formed with granular center and jagged edges, which resembles daisies. The principles of microbiological diagnosis of plaque include bacterioscopic, bacteriological, serological, and biological research methods, which are carried out in special laboratories licensed to work with pathogens of especially dangerous infections. The research material are buboes, the contents of ulcers or other skin lesions, sputum and mucus from the throat and blood. During bacterioscopic examination, the prepared specimen is fixed in Nikiforov's mixture and stained according to Gram or methylene blue according to Leffler. Immunofluorescence reaction is used as an express diagnostic, which makes it possible to make preliminary diagnosis after 2 hours. The direct immunofluorescence reaction method is based on the fact that tissue antigens or microbes treated with immune sera with antibodies labeled with fluorochromes are able to glow in the UV rays of a fluorescent microscope. Bacteria in a smear treated with such luminescent serum glow along the cell periphery in the form of a green border. In a bacteriological study, a pure culture is isolated and identified. For this purpose, the test material is sown on nutrient media. An effective way to isolate a pure culture of the plaque microbe from a material contaminated with foreign microflora is a bioassay on guinea pigs and white mice. Serological examination is carried out by staging, indirect hemagglutination reaction, linked immunosorbent assay, pH antibodies. As an allergic diagnostic method, a skin test with spestin is performed. An allergic test is a method of retrospective diagnosis. Plaque bacteriophage is also used in the diagnosis of plaque. They are filtrates of plaque bacillus phagocytates with specific lysing activity. The lytic activity of the bacteriophage is determined by the Appleman method with a reference culture of the vaccine strain and with 7 virulent strains of the plaque microbe isolated from different rodent species. The bacteriophage can be released if all 8 strains are lysed and 4 of them have titer of at least 10 in the power of -8. Specific prophylaxis is carried out by a leaf attenuated vaccine based on the EV strain. Vaccines are used according to epidemic indications in epidemic foci. After vaccination develops immunity lasting up to 6 months, the vaccine is administered once using a needleless injector. A tableted leaf vaccine from the EV strain for oral administration, as well as vaccine for inhalation use in emergency situations, has been developed. Non-specific prevention of plaque is of great importance, which includes the following measures: prevention of human disease and the occurrence of epizootics in natural plaque foci, prevention of the importation of plaque into the country from other regions through the implementation of special international sanitary rules, Also, the prevention of infection of persons working with infected Yersinia pestis material carried out by the regulation of the work of anti-plague institutions. Tularemia is a zoonotic natural focal disease that occurs with intoxication, fever, lymphadenitis, damage to various organs, a varied clinical picture, and slow recovery. The name of the disease and the specific epithet of the pathogen came from the province of California, Tulare, where the bacterium was first isolated from ground squirrels by researcher McCoy and Chapin. The genus name comes from the surname of Edward Francis, a researcher of Tularemia. The microorganism is assigned to the second group of pathogenicity. Francisella tularensis, a species of small gram-negative bacteria with polymorphism, do not form spores, form pseudocapsules, immobile facultative intracellular parasite, meaning that they can survive inside and outside of the cell. Antigenic structure have two antigens. First, O-somatic, cell wall lipopolysaccharide. There are common substances with Brucella antigen, which can lead to cross-reactions. Second is VI antigen, which is capsular, is released only in virulent forms of Francisella in the S form of the colony. Pathogenic factors. The causative agent of tularemia is an intracellular parasite. Virulence is due to a capsule that inhibits phagocytosis, neuraminidase, which promotes adhesion, endotoxin, allergenic properties of the cell wall, the ability to multiply in phagocytes and suppress their killer effect, the presence of receptors for the Fc fragments of immunoglobulin G to suppress the activity of complement systems and macrophages. Francisella tularensis doesn't grow on normal media, requires cysteine-enriched media for growth, grows in 47 or 72 hours. Chocolate agar, named for its color, contains cysteine and least red blood cells. BCYE medium is a buffer charcoal yeast extract that contains activated carbon, yeast extract, and L-cysteine. Francisella tularensis forms round gray-white colonies on these two media. CHAB environment is contained glucose cysteine, thiamine, blood. Francisella tularensis produces greenish-white, round, smooth S-shaped colonies on this medium. On liquid media, growth is superficial. They break down on some carbohydrates with the formation of acid, form H2S. Laboratory diagnostics. Materials: pus from buboes, eye discharge, punctate of lymph nodes, sputum, blood, and sectional material. Basic methods. First is allergic, which uses a test with tularine. Second is serological, which starts from the second week of the disease that uses indirect hemagglutination reaction, and blood drop reaction. Third is bacteriological, that assesses the nature of growth on nutrient media and lack of growth on simple nutrient media. Fourth is express method, that searches for microbial antigens, that uses immunofluorescence reaction, linked immunosorbent assay, and polymerase chain reaction. Fifth is biological. Biological assay as the most sensitive way to detect tularemia bacteria in any test material. Prevention. Specific prophylaxis according to epidemiological indications in outbreak. The Albert Guyski vaccine is a live, dry, attenuated vaccine, administered once, skin or intradermal. Microbiology of cholera. Cholera is a disease that is characterized by toxic damage to the small intestine, a violation of the water-salt balance, dehydrating diarrhea with stools in the form of rice water. Taxonomy. Family of Vibrionaceae, genus Vibrio, species Vibrio Cholera, Vibrio Cholera asiatici, which is classical, and Vibrio Cholera alter and 3 serotypes. Morphology. Slightly curved or straight gram-negative rods. Spores, capsules do not form. Mobile due to a long flagellum at the end of the cell. Facultative anaerobes with aerophilic properties. long remain in reservoirs, food products. Antigenic properties. Vibrio Cholera has somatic O antigen and a flagellar H antigen. According to the structure of the O antigens, they are divided into 139 serotypes. The causative agents of classical cholera, Vibrio Cholera asiatica and Vibrio Cholera alter, are combined into serogroup O1. Within the O1 serogroup, depending on the combination of A, B, and C subunits, three serotypes are distinguished. The combination of A-B subunits is called the Ogawa serotype. The combination of AC is called the Inaba serotype, and the combination of A-B-C is called Gikoshima. The last serotype is transitional between the first two. Many currently dispute its existence. NAG, non-agglutinating vibrios, non-pathogenic and opportunistic Vibrio Cholera not belonging to serogroup 1. Cultural properties on solid media: S-shape, smooth, flat colonies, grows after 18 hours. Cell division cycle 15 minutes, which is short. On alkaline agar, pH 8.5, colonies are smooth, which shows transparent, viscous consistency. In transmitted light, a grayish-bluish tint. On TCBS, which is thiosulfate citrate bromothymol sucrose agar plus bile salts, Vibrio Cholera ferment sucrose. Growth of yellow colonies on a bluish-green medium. In liquid medias, on 1% peptone water after 8 hours, a bluish-gray film, soldered to the edges of the test tube. The broth is transparent. When shaking, the film breaks and settles to the bottom of the tube. Proteolytic properties. Liquefied gelatin forms indole. Saccharolytic properties are determined in relation to three carbohydrates, which are sucrose, arabinose, mannose, that are known as Heyerbeck's triad. Breaks down sucrose, mannose to acid, however doesn't break down arabinose. Research methods are divided into bacterioscopic, bacteriological, as well as express diagnostics, which include enzyme immunoassay, immunofluorescence reaction, and polymerase chain reaction. Bacteriological method. The material for bacteriological analysis can be feces, vomit, bile, objects contaminated with feces. The material for research should be delivered no later than 2 hours after it was taken. In the case of lengthening the delivery time, transport media are used. The most convenient and quite effective is 1% peptone water. Material research is carried out according to the following scheme. Stage 1 includes microscopy of smears from pathological materials such as gram stain, magenta, and the drug crush drop. Sowing the material on the enrichment medium, 1% peptone water. Sowing the material on alkaline mid-peptone agar and differential selective media such as TCBS, TTGA, etc. Stage 2 is carried out in 6-8 hours. At this stage, the nature of growth on the first peptone water is taken into account, which is delicate blue film. Next, microscopy of native and fixed smears from the film is carried out. Gram stain, fusion, determination of mobility. After that, reseeding is done on the second peptone water for further enrichment, and also transferring from the first peptone water to alkaline agar and TCBS medium. Stage 3 is carried out in 12-40 hours. Growth is recorded on the second peptone water, which is similar to the first. Studies the nature of growth on plate nutrient media. Carry out microscopy of smears stained by gram and magenta from yellow colonies on TCBS medium and transparent ones on alkaline mid-peptone agar. Determine the mobility in preparations crush drop. Put the agglutination reaction on the glass with O1, Inaba, Ogawa, and Arosera. Next, reseeding of agglutinable colonies on Klager's medium is done. At this stage, a tentative answer may be given. Stage 4 is carried out after 18-24 hours. Record changes in the lactose-sucrose environment. Identification of the selected culture is carried out including extended agglutination reaction with Cholera O1 Inaba Agawa Ero. Also, determination of sensitivity to cholera diagnostic bacteriophages and determination of belonging to the Hairbeck biochemical group. In addition to that, definition of biovar and determination of the spectrum of antibiotic resistance. Methods of prevention. Non-specific. Compliance with sanitary and hygienic measures. Specific ones. The following types of vaccines have been proposed to create artificial immunity. First, corpuscular killed vaccine. Second, choleragen toxoid. Third, bivalent vaccine consisting of toxoid and somatic antigens, Inaba and Ogawa serotypes. Fourth is live oral vaccine. Emergency prevention of cholera is carried out by mass prophylactic administration of tetracycline in areas of epidemic danger. Other antibiotics effective against Vibrio Cholera are used for the same purpose. Thank you for your attention.